Hello, my dear students. Now we are going to discuss some important terms frequently used in genetics, because it will help you to have some better understanding of the genetics. And we will start by by the term gene. Gene is the most important and most common term used in genetics, and you can say that genetics essentially revolves around this term, and that is why some have defined genetics as a study of the gene. This term was first introduced by Wilhelm Johansen in 1909. You will be a little bit surprised to know that the father of genetics, that is Mendel, did not know this term, because it was first given by Wilhelm Johansen some 25 years after the death of Mendel. So, what is gene? How can you define it? We define this gene as per our requirement. For example, as in classical genetics, the focus of our study is on characteristic. When the characteristic finally appears in an individual, when it is expressed in an individual in a stable form. And when the characteristics appear in an individual and we follow this characteristic from generation to generation, we follow these characteristics, appearances and disappearances, or variation in the degree of this expression. And as the characteristics are defined by this, controlled by this gene, accordingly we follow actually the transfer of this basic unit, that is this gene, from generation to generation by studying the character for this appearances and disappearances of the characteristic. So here our main focus is on the characteristic of an individual and how this characteristic phenotypic feature, which can be easily observed by an observer, how this phenotypic feature is moving, is getting transferred from the generation to the generation, and accordingly we can predict the transfer of this gene. And in classical genetics we accordingly define it as the inherited factors which determines a characteristic, that is a characteristic which appears in an individual or which is expressed in an individual is because of factors, because of some physical entity which has been inherited from its parents. This is how we define in classical genetics. In classical genetics, we simply say that this gene is some kind of physical factor, physical entity which controls a characteristic in an individual, or you can define it in another way, that it is a unit of heredity which gets inherited from the parents and controls a characteristic, phenotypic feature in an individual. This is in the classical genetics, but in case of the molecular genetics, requirements are different. In molecular genetics, you are supposed to explain how the genetic information is encoded within this gene and how this information gets translated from this gene into the protein, how it is expressed. Now the requirements are different, so we define this gene in different way. In molecular genetics, we define it as sequence of nucleotides essential for the synthesis of gene product. We define it as the gene as a sequence of nucleotides which are essential for the synthesis of gene product. Gene products are either, it can be the RNA or it can be the proteins. Let me explain it. First, you have to understand what does it mean by the sequence of nucleotides. This term, this nucleotides, it will give the chemical nature of the gene. That is, this term nucleotide, it says that gene is composed of nucleotides. That is chemical nature of the gene. That is not, it is not composed of the amino acids or carbohydrates or the lipids. It is composed of nucleotides. These nucleotides get linked together through phosphodiester bonds, and they will form the nucleic acid molecules. Chain of these nucleotides, they form these nucleic acids. That is, you can say that this gene is actually the nucleic acid. That is, it is the sequence of nucleotides, that they are composed of, their composition, this molecular composition is nucleotides. As you know that these nucleotides have one phosphate group, then another is sugar attached with nitrogen base. This is the basic building block of gene. That is, this is called as nucleotide. That is, in a single nucleotide molecule you will find a phosphate group, a sugar, a nitrogen base. This sugar can either be the ribose or deoxyribose. And nitrogen bases, as you know, we have 5 most common nitrogen bases. For example, we have this adenine, then we have guanine, cytosine, thymine, and in case of this RNA, we have uracil. This nucleotide is composed of 3 molecules. That is, single molecule of the nucleotide is basically composed of the 3 molecules. One is this phosphate group, then we have this sugar, then we have the nitrogen base. Nitrogen base plus sugar plus phosphate forms one nucleotide molecule. As you can see, this sugar can either be the ribose, having OH group at the second carbon of this sugar, or it can be the deoxyribose. Nitrogen base can either be the adenine or guanine or cytosine, thymine, or this uracil. So this is the structure of the nucleotide. That is, molecular constituents of this nucleotide or phosphate, sugar, and the nitrogen base. And here we are saying that this gene is a sequence of nucleotides. That is, gene is composed of basically nucleotides. These nucleotides, this molecule of the one nucleotide molecule will get linked with another molecule of nucleotide through phosphodiester bond, and in this way these nucleotides will form a chain of the nucleotide, which is called as the nucleic acid. And we have 2 types of nucleic acid. One is called as the RNA and another is called as the DNA. This classification is based on kind of this sugar. Here, if it is ribose sugar, then it is the RNA. If it is deoxyribose sugar, then it is the DNA. So basically, the gene, it is a sequence of nucleotides. So this is about the chemical nature. That is, gene is essentially composed of nucleotides. Second important thing you have to understand is that it is this sequence. What does it mean? It means that nucleotides, when they are present in a sequence, that is, any change in these sequences of the nucleotides, as you know, we call it as a mutation. And this mutation in some cases are having some serious complications so far as the characteristics or the expression of this gene is concerned. That is, nucleotides, when arranged in specific sequence, only then they will be called as the gene. That is, just having these nucleotides is not enough for the nucleotides to get qualified as a gene. These nucleotides must be in some sequence. For example, you must have heard about the sickle cell anemia. This sickle cell anemia is, as you know, is a genetic disease. And in this sickle cell anemia, we have abnormal hemoglobin. and that abnormal hemoglobin we denote it by HB, the S, to differentiate it from the normal hemoglobin. And this sickle cell anemia is a result of a point mutation. I am just trying to explain how important this sequence is. If there is any change in this sequences, it will in many cases have some serious consequences for the survival of an individual. So here in sickle cell anemia, we have abnormal hemoglobin, and accordingly we have some complications in the individual in which this mutation occurs. That is, this individual will be having low count of RBCs, flexibility is, they are not so flexible, those RBCs, and we have this sickle shaped this RBCs. So these are some phenotypic features which have some serious complications in the individuals. What is the cause of these phenotypic features is a point mutation. This is normal gene. Normal gene is having sequence this G, A, and A. There's a guanine, adenine, and then we have this again guanine. Here in this case a mutation occurs and this adenine gets changed into thymine. The gene which encodes the beta globin chain of the hemoglobin, in normal case it has a sequence like this: G, guanine, adenine, and then guanine. And this sequence, when gets translated, forms an amino acid that is called as glutamic acid. This is normal hemoglobin gene. At the 6th position, we have this glutamic acid, and we have sequence like this: guanine, then adenine, and guanine. Now, a point mutation occurs, that is, a single nucleotide is here changed, and it forms this thymine. Now, when this thymine is introduced here, after translation here, it will be forming valine. Now, first in the normal hemoglobin, we have glutamic acid at the 6th position of beta chain. And when point mutation occurs, now we have valine at the 6th position. Now, you can say that after mutation, a valine replaces this glutamic acid. This introduction of valine at the place of glutamic acid changes the properties of the hemoglobin. As glutamic is hydrophilic amino acid and valine, it is a hydrophobic, and these change in the these properties of the hemoglobin causes the secondary consequences like loss of flexibility of the RBC, the lifespan of the RBCs gets declined, and in some cases it causes the blockade of the blood vessels. So these are the complications associated with this sickle cell anemia. But basically, how does it occur? The basic reason, basic cause is that the single nucleotide here, that is adenine, it gets changed to the thymine. That is, here I am trying to explain the sequence. That is, just presence of this nucleotides is not enough, is not sufficient. What we should have these nucleotides in a specific sequence. If there is any change in that sequence, it will lead to the mutation, and these mutations, although in majority of the cases these mutations remain silent, but in some cases they will be having some serious consequences, serious complications, and they will definitely affect the survival of an individual. And here you can take example of this sickle cell anemia, that is replacement or the change of the single nucleotide synthesizing an abnormal hemoglobin chain, and that abnormal hemoglobin chain having the serious consequences for the individual in terms of the phenotypic features. So here gene, chemically it is of nucleotides, but these nucleotides must be in a specific sequence. Now, another important point, in living organisms we have both coding sequences and non-coding sequences. Coding sequences are those which are being translated into the proteins, and non-coding sequences are not translated into the proteins. For example, in case of human beings, we have almost 6.4 billion base pairs. 1% is getting translated into the proteins. Some have called the remaining 99% as a junk DNA. So can we include those non-coding sequences in the definition of gene? Answer should have been no. But there are some sequences which are included, which are non-coding, that is, which are not getting translated into the protein, but we are including them in the definition of the gene. For example, if you take the structure of the gene, here you will find the enhancer, then you will find the promoter, then you will find the main transcribing region, then you will be having this terminator. And only this transcribing region, this one, is getting translated into the sequence of amino acids or RNA. And rest of these nucleotide sequences which are present in this enhancer region or in promoter region or terminator region, these are non-coding sequences. That is, a gene structure is having first these non-coding sequences in the enhancer region, then in the promoter region, and also in the terminator region. But this enhancer region, this is short segment of the nucleotide sequences which enhances the transcriptional activity of the gene. And similarly, in the promoter region, we have nucleotide sequences where first the transcription factors get attached, bind at this region, and then those transcriptional factor proteins, they recruit the RNA polymerase, and then RNA polymerase synthesizes the starts the synthesis of the RNA chain. Again, the sequences in this promoter region are non-coding. And we have this terminator region where we have the nucleotide sequences. These nucleotide sequences signal the release of newly synthesized RNA strand and release of this transcriptional machinery. So we have non-coding nucleotide sequences in the enhancer region, then we have in the promoter region, then we have terminator region. And only this transcribed region, only in this region we have nucleotide sequences. These nucleotide sequences are getting translated into the protein as a sequence of amino acids or into the RNA as a sequence of the nucleotides. But these three regions are non-coding. Here I am telling you that the sequences which are non-coding but they are essential. That is, this sequences present in the enhancer region, promoter region, and terminator region. Although these sequences are not by itself getting translated into the protein or RNA, but the presence of these sequences is essential for the activity of the gene. That is, a gene cannot function without having these non-coding sequences. So, in the definition of gene, we are including coding sequences as well as some non-coding sequences. Those non-coding sequences are essential for the activity of the gene. Although those sequences are not by itself getting translated into the sequence of amino acids, but their presence is important for the functioning of the gene. So, we have sequence of nucleotides. That is, basic chemical nature of the gene is nucleotides, and these nucleotides should be present in some specific sequence. Change in sequence can usually affect the survival of an individual. Then, are all nucleotide sequences essential? No, only those sequences which can affect the functioning of the gene. Coding sequences by itself are essential, and some non-coding sequences are essential which directly affect the activity of gene are included in the definition of the gene. So, sequence of nucleotides essential for what? Essential for the synthesis of gene product. Now you can define gene as a sequence of nucleotides. That is, nucleotides present in specific sequence, essential. And in essential, we can have the nucleotide sequences which by itself are translated into the sequence of amino acids, or in the sequence of nucleotides in the RNA. And also those sequences which by itself are not translated into the protein or this RNA, but their presence is essential. They have only one function, that is to synthesize gene product. That is, only those sequences which are involved or which get translated into the gene product, and this gene product can either be it can be either RNA or it can be either this protein. For example, in majority of the cases it is protein. That is, in majority of the cases, gene forms protein as the final functional product, and it is the protein which finally performs the functions as per the instructions. That is, a protein is the final functional product which performs the function as per the instructions given in the DNA or gene. But in some cases, RNA can act also as a final functional product. That is, once the RNA is formed from the DNA, this RNA, after some structural changes, can perform functions. For example, we have this ribosomal RNA, or we have this transfer RNA. That is, these two molecules are RNA molecules, and these RNA are not getting translated into the protein. For example, in normal cases, we have the flow of genetic information flows in this direction. Here you can also include the reverse transcription, where RNA forms the DNA. Here DNA forms the RNA, and then RNA forms the protein. And in some cases, RNA does not form the proteins. It remains as RNA. And as in the form of the RNA, it performs various functions. And this RNA, after getting some modifications, post-transcriptional modifications occur in these molecules, and after that they perform various functions in the cytoplasm. And these RNA molecules are as the functional gene product. So, in this definition, we have gene as a sequence of nucleotides which is essential for the synthesis of gene product. That is, genes are chemically nucleotides. They are composed of nucleotides. And these nucleotides, when arranged in a specific sequence, and then all those sequences which are essential, whether they are encoding, whether they are getting translated into the protein or not. If the non-coding sequences are essential for the activity of these proteins, then those non-coding sequences are also included in the definition of the gene. And this sequence of nucleotides, which is essential for the synthesis of gene product, and this gene product can either be the RNA or it can be the protein. So this is the definition of gene.